to Brandeis, located 9 miles from downtown Boston in Waltham, Massachusetts. Brandeis combines the intimacy of a small and interactive research community with world-class science faculty, research facilities, and graduate programs. Brandeis' collaborative environment, philosophy of inclusion, and focus on mentorship makes it a unique place to launch your scientific career. Scan these QR codes if you'd like more information about the biochemistry and biophysics, molecular and cell biology, and neuroscience PhD programs. To give you a feel for the scientific community at Brandeis, we're going to take you on a virtual tour to meet some of our current life sciences doctoral students.
Hi.
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 Hi, my name is Dr. Vic Sarkisian.
My name is Raul Ramos.
Hello, my name is Tyler Hill. I'm a fourth-year graduate student in the lab of Piali Sengupta.
I'm Ingrid. I'm a fifth-year graduate student in the Curran lab.
I'm a fifth-year graduate student in the Garrity lab here at Brandeis.
Hi, my name is Elias Vanvakis, and I am a second-year PhD student in the Rodel lab.
I am a sixth-year neuroscience PhD student in the Terrigiano lab.
I'm Alexis. I'm a third-year MCB student in the Bradshaw lab. So welcome to our brand new lab space over here in Bolin.
The reason I came to Brandeis is because of, one, the world-class research that comes out of Brandeis.
I joined the Terrigiano lab because I was really interested in how synaptic plasticity was important for learning and memory.
One thing that I really love about Brandeis is from the very beginning, its historical mission has been to show the world that strength comes from diversity, and that by opening up your arms to all the wonderful people in the world, that you can create wonderful ideas and share all of your diverse experiences. And that's something that's really important to us.
There are a lot of reasons why I chose Brandeis, but I think the most important one is that they have this really great umbrella program. So if you're an MCB student, you can rotate in either MCB, neuro, or biochemistry labs, any of them within the department.
One of the things that I really like about Brandeis is the collaborative nature amongst all the labs here. So what that means is that often students will end up on projects that are in collaboration with 2 or sometimes 3 labs.
It possesses the kind of rigorous research program and highly collaborative environment necessary for a great doctoral training.
A lot of the work here has managed to really spark entirely new fields of inquiry, and I really see that as a rising out of these intimate interdisciplinary interactions. So just think about Jeff Hall and Michael Rosbash and sort of taking behavioral genetics and molecular biology and turning it into this field of circadian rhythms. I think that's just a beautiful example where these close personal interactions really just gave rise to something new. Think about computational neuroscience and some of the really seminal work that was done here arose out of the same thing, physicists talking to neuroscientists. And I think there's just lots of examples of that. So I see it as just being part of a very close-knit community, but also I think the people here are a little bit self-selecting for people who are really broad-minded and like to think across boundaries. We don't see the boundaries, we see the connections.
Our university motto is truth unto its innermost parts, and so I love that we really value the deep dive,
Yes.
Really getting to the bottom of things. And so when you combine that broad-mindedness with really caring about understanding at a deep level how things work, then you can really do some awesome stuff.
I really chose to come here because of the faculty and also the students.
For me, Brandeis felt like a natural fit because it had the research capacity of a large university without any of the drama.
I came out of fairly small schools, and I knew I didn't really want to jump into a huge research university, something that was very impersonal.
A lot of the PIs here at Brandeis, especially since we're so close to Boston, are really well connected with professors at other universities.
When I came to visit Brandeis, I found that there was a lot of collaborative nature between the labs. Everyone seemed to know each other. Everyone was very friendly with each other.
We all talk to each other. We all work together to solve problems and to get answers that we want to know.
It's just been a very supportive, very helpful environment where I've been able to work with my peers and my PI to be well on my way to being an expert in a topic that was completely alien to me when I started.
My PI is really great about supporting the interdisciplinary opportunities to work on your research. So I'm able to take a lot of different approaches to answer the questions that I'm interested in.
I knew instantly that I would choose to start my PhD here. I saw immediately that everybody, the students and the professors, were so passionate about their research.
Faculty are very approachable and accessible.
A lot of the labs had papers with other members of the Brandeis community, and I was just really interested in becoming a part of that.
It's important to say that we really value scientific rigor and teaching students how to think deeply and clearly and how to approach problems with the appropriate quantitative tools. I mean, all of that is really embedded, I think, in the way we do science.
So currently, I'm working on a project where we study the role of homeostatic synaptic plasticity in associative learning and memory. My thesis project is a collaboration between our lab and the lab of Don Katz. His lab specializes in the study of taste behavior and taste learning, and we are using techniques that we have established in our lab for synaptic interrogation and for understanding synaptic plasticity to understand how the specificity of taste memories is encoded at the cellular level.
In the Garrity lab, we study the ways in which insects such as flies and mosquitoes detect temperature and humidity. Mosquitoes in particular use specialized sensory cells to locate and bite human hosts. So understanding the ways in which sensory receptors mediate the detection of humidity and temperature can help us prevent the spread of diseases such as malaria and Zika.
My research is focused on understanding the mechanism of allosteric regulation of the protein tyrosine phosphatase SHP2. SHP2 is a big player in signaling pathways, but naturally it's inactive, it's off. So nature developed a way to control this phosphatase's activity, and it does so by the allosteric regulation using two SH2 domains. Now, these domains lock the phosphatase in a closed conformation, but how does it work? How can it be turned on again?
Our lab works principally on how cells employ membrane remodeling to form complex structures to fulfill equally complex tasks. So one of the main tools I use in this process is this spinning disc confocal microscope, which we call Jabberwocky, which allows us to see in very close detail what's going on in the synapse and with our proteins of interest.
I work on thermal sensation in the nematode C. elegans. One thing that's really interesting about C. elegans is that unlike many other organisms who really, they want to be at one set temperature, their bodies and their behavior really moves them towards that one set temperature. But with C. elegans, the temperature that they move towards is actually dependent on their temperature experience. So if you grow them up in colder environments, they actually want to move towards colder environments. If they grow them up at warmer environments, they really want to go to warmer environments. And we really don't know why that is. The temperature sensing neurons in these C. elegans worms actually respond to a specific temperature that is really highly correlated to the temperature that we cultivate them at. And what I have found is that this shift actually happens very, very fast. If you expose these worms to a higher temperature for 1 or 2 minutes, you can see that the temperature that these neuron responds at it shoots up. It moves towards that higher temperature really, really fast. And we just have no idea how that works, it's really interesting to me. That's why I got into science, is to figure out things that no one else knows, just because I could be the one to figure that out.
Overall, my project focuses on how signaling pathway proteins overlap with cytoskeletal machinery in bacteria. So what I've been looking at is how a signaling phosphatase called SPO2E is involved in manipulating the bacterial version of tubulin to promote sporulation and downstream programming that allows them to form these really stable endospores. And so far what I'm finding is that my phosphatase does seem to work with this machinery. So what I'm doing is a lot of single molecule analysis to figure out like exactly what the effects are of my protein on tubulin. So I know it's probably hard for you to make this decision without actually being here on campus with us, but I guess if there's one thing I could convey that I got out of in-person interviews that you might not get to see, is that graduate students really are a family. This place is a home. The PIs they're real people, but everyone's also really excellent scientists, and they're really interested in making sure that you're able to have the successful scientific career that you want.